Very good morning, students. So this is the part of the previous lecture that we had taken up for the types of the heart on the basis of the morphology of the hearts. So in the previous lecture, we had discussed about the very first type of the heart on the basis of the morphology, and that was the chambered hearts. Now here, what we have is the second type. So here we have the second type of the heart, which is based upon the morphology, and this type of the heart is the tubular hearts. So this is the second category which we have in the types of the heart. Now students, if we take these hearts into account, so these hearts, they consist of long tube-like vessels. So these are long hearts, these are tube-like hearts. The systemic hearts of most of the arthropods, they consist of these contractile tubes. So here I am taking the example of the Limulus in this particular type of the heart. Now there may be a very thin kind of receiving chamber which is very equivalent to the atrium in this particular case. So as the atrium is the receiving chamber is in most of the animals, as it is not muscular enough to pump the blood, but it is receiving or you can say primary storage of the blood, as we have seen in the case of even the fishes. So here there can be a thin receiving chamber which is equivalent to the atrium in this particular animal, the Limulus. So it is surrounded by a particular, you can say, the cardiac ganglia, as it is a neurogenic kind of a heart. So the surrounding part of the heart, as in the case of the Limulus, or the heart, it may be free in a large sinus in this animal. So what is surrounding this particular heart is the hemolymph, and that is inside a sinus or an open space which is called as the pericardial sinus. Now this kind of heart, it is long, as you can very well see here in this diagram. It is tubular, as we have taken it in the tubular heart range. So it is usually anchored at several corners with some muscles, and it is receiving the blood through several paired aortae which are valved. So they have got valves. So here you can have the ostia. So these are the ostia over here which are present. These are the ostia from which the blood will enter into the heart. So these are very equivalent heart to the cockroaches, which are the next taken in the next slide. So if I go further after Limulus, we have the example of the cockroach in the tubular hearts. Now what we have in the cockroach is that there are many chambers also. So this type of the heart is also considered to be as the chambered heart, as we have 13 chambers in total in this particular heart. So the heart is suspended by various Ampullary muscles, which you can see over here. So these are the Ampullary muscles which are anchoring this heart in the body of the cockroach. So here you can see the chambers of the heart. So it is having 13 chambers in all. If you take the last segment of the heart, it is the 13th chamber. So after going into the chambers of the heart, what we have is that the Ampullary muscles, they are maintaining this heart in the particular position. The blood, it leaves the heart by one or several arteries, which is typically at the anterior end. And here you can very well see the presence of the anterior aorta, which is the main vessel which is taking the blood to the head side. So the you can say the myographic or the ECG type of the graphs of this particular heart, they indicate that the entire tubular heart of the animal, it is contracting in a wave-like manner. So a wave of contractions, it is occurring in this particular heart. So what is important over here is the presence of the ostia. So here you can see this is an ostium which is present, and the blood, it can like move into the ostium from here if it is open. So it will reach the chamber. So when this particular chamber has to contract, what will happen? These ostia will be closed, and these ostia now are closed. When these are closed, this particular chamber will contract, and the blood, it will flow into the second chamber and then into the third chamber, and so on. So ultimately what happens, that the blood will flow out of this particular aorta to the head side. So this is the mechanism in the case of the cockroaches. Now students, after the tubular hearts, now we have the very third type of the heart, which are the pulsating vessels. They are pulsatile in nature, and they are small vessels, they are blood vessels which are pulsatile, and they are showing the peristaltic movements. That's why they are taken as the heart. So the blood vessels which contract with the peristaltic, you can say, wave-like motion. So they are widely distributed in many of the animals. So in the annelids, many blood vessels, they show rhythmic peristaltic movements. So I am taking here the example of the earthworm. So in the earthworm, the contraction waves in the dorsal vessel, they pass from the posterior to the anterior end at a very high speed, and that can be like 20 millimeters per second, and the waves are repeated at the rate of about 15 to 20 per minute. So this is a kind of, you can say, the speed of the waves in the case of the earthworm. The lateral vessels of the earthworm, they usually refer to as the hearts. So here you can very well see these are the two types of the heart that are present in the earthworm. Number first heart, if we take the left diagram, this is the lateral heart, and if you take the second diagram, it is the lateroesophageal heart. So here, what is the difference? That if you see the base of the heart, it is connected to the ventral vessel. So that is the lateral heart, and on the top, it is connected to the dorsal vessel. So it is connecting the dorsal vessel with the with the ventral vessel. But if you take the second diagram, it is connecting the dorsal vessel from the top, and it is also connecting the supraoesophageal vessel with the ventral vessel. So this is the main difference in between the lateral and the lateroesophageal hearts. Now these hearts, they contract at the rates differing from that of the dorsal vessel. So they have got their own contraction mechanisms, and they are contracting at different rates from the dorsal vessel. Each heart, it beats at its own rate. Now there is no similarity in between the beat rates of these two types of hearts in earthworm. They are beating at separate rates. Although the two hearts of a segment, they tend to contract together. As the earthworm has got 4 pairs of hearts, so a pair in a single segment, they are contracting together, but they have got no relation with the other segment, you can say, the hearts of the other segments. So that was the case of the earthworm. Now next we are taking the example of the leech. Now in the leech, instead of a dorsal vessel, there are 2 lateral vessels which contract alternatively. So this is the diagram, and here you can see these are the heart tubes. So these are the 2 heart tubes which are contracting alternately, and if you take the case of the nereis even, the dorsal vessel contracts rhythmically at a somewhat slower rate than in the earthworm. So in the case of Nereis, you have the dorsal vessel, but here in the case of Leech, we have got the lateral heart tubes or the lateral vessels which are acting as the heart. Now the blood flows forward in the dorsal vessel in case of Nereis and passing on the each side of the gastric plexus to the lateral gastric vessel and hence to the lateral heart which pumps the ventral vessel. So the area connecting the lateral gastric vessel to the heart, it beats before the heart. And sometimes the several time beating during a single contraction of the heart. So the dorsal and the lateral vessels, the esophageal vessels, and some nephridial vessels, they are contractile also. So in general, if we talk about the annulates, so the contractions of the pulsating vessels in annulates, they are not as regular as the beats of the chambered hearts. So here you can see the heart tubes. In the previous section you had seen the lateral and the lateral esophageal hearts of the earthworm. They beat like a heart, but they are not beating as the chambered heart. So they are less efficient as compared to the chambered hearts. Now students, after this we are taking up the very fourth type of the hearts, which are the ampullary hearts. Now this is the fourth type of the heart based upon the morphology. Now in several animal groups there are these accessory organs or the ampullary hearts present, and they propel the blood through the peripheral channels to the heart. Now students, this is a very, very important thing, that ampullary hearts are the accessory organs which are present along with the heart to push the blood through the peripheral channels. Now here I take up the very first case of loligo. Now this is the first example. Now here the branchial hearts of the cephalopods, they consist of a spongy tissue of some kind of endothelial cells surrounding many small vessels. So this is the diagram of branchial hearts of the loligo. So here you can very well see that this is the gill. If you see this particular structure, this is the gill, and here we have the branchial heart. Now there are two branchial hearts which are persisting in the case of loligo near to both the gills. Now one branchial heart is on the left side and one is on the right side. So they are acting as accessory organs for propelling the blood through the peripheral channels. Now these hearts, they are interposed between the systemic veins and the gill arteries. So here you can very well see that this is the atrium and the ventricle in the very center. This is the heart of the loligo, and these are the accessory chambers which are giving blood to the gills for you can say oxygenation. So that was the case of loligo with ampullary hearts or the branchial hearts. Now students, we are taking up the case of the insects. Now here the accessory hearts, they are very common and they are particularly present at the base of the antenna. So here you can very well see this is the ampulla, the number 5. This is the ampulla of this particular heart, and this is the antennal vessel which is going into the antenna of the insect. Now at the attachment of the wings and the legs, they are also present. So there can be many accessory hearts which are present in an insect. So the position can be at the base of the antenna, at the attachment of the wings and in the legs. So insect accessory hearts, they consist of some muscle fibers arranged in various patterns, and they serve to keep the hemolymph moving in the open circulation. So as we have sinuses in case of the insects, there is an open circulatory system. So these types of hearts, they keep on the hemolymph moving inside those sinuses. So this particular diagram, if you see this diagram, the antennal circulatory organs or the ampullae, they have been shown. So here you can see the ampulla, which is the antennal circulatory organ. Now this figure, this diagram, it also shows the head diagram in the dorsal view of the insect. Now the vessels and their derivative structures, they are shown in solid black color. So here you can see the ampulla in a solid black color. The arrows, they indicate the hemolymph flow. Now here you can very well see that the hemolymph is flowing first of all to the upper part, then it is moving on to the lateral sides. So when it is reaching the lateral sides of the head, this ampulla will pump the hemolymph into the antenna. So these are the kind of accessory, you can say, pressure making chambers. So the circumoesophageal vessel ring with the ventral trumpet shaped opening carries the hemolymph, which enters the ostia on both the sides. So here you can very well see in this particular diagram that the hemolymph, it is entering into the head part near the ampulla. So ultimately it enters the ampullae, which pump it to the antennal vessels. So we can see the antennal vessel also. So this is the flow of the hemolymph in the case of insects with the help of these ampullary hearts. Now students, after the insects, we are talking something about the Lepidopterans. So Lepidoptera, it is the order of the butterflies and moths. Now here, what is happening, that the hemolymph pumping organs, the organs which are pumping the hemolymph, they have been found in these types of the insects also. It has been seen to be present in the Lepidopteran proboscis, and they are involved in the hydraulic extension of the proboscis. As the butterflies, they are having a siphoning type of mouth part, and they have a round coiled kind of a proboscis. And if we go for the extension mechanism of that proboscis, these hemolymph pumping organs, they are helping in the extension of the proboscis in the Lepidopterans. Now these organs, they are located near the proboscis base, and they consist of cuticular tubes with associated muscles. So the contraction, it presses the tubes together, whereby the hemolymph is forced into the proboscis. So with the muscular action of these organs, the hemolymph, it is propelled, it is pushed into the proboscis tube. So ultimately that elongates the mouth parts of the insects by special circulatory organs for the hemolymph exchange in the case of Lepidopterans. So that was the case of the butterflies. And if I go further, we have some case in Orthopterans and Hemipterans. So Orthopterans are the hoppers and Hemipterans are the flies. So here also we have the pulsatile organs in the legs, and they are well known in the Orthopterans and the Hemipterans. They are present in the legs. Now in these insects, a delicate, you can say a non-muscular diaphragm, it takes over the channeling function for each extremity of the leg. Now here in this particular diagram, you can see that where is the sinus? This is the sinus. So here is the sinus number 1. It has been shown as white in this particular thing. And here we have the sinus number 2. So this is shown as the black sinus. And these sinuses, they are present in the legs of the hemipterans or the arthropods. Now, it longitudinally spans the entire leg of these insects and ends before the leg tip. So if I talk about this particular channeling, so it is in the entire leg. So here you can see the channel of the sinuses, and you can very well see the mechanism of the flow of the hemolymph. So here you can see that the hemolymph is coming down from the femur into the sinus number 2. It is flowing into sinus number 2, it is going down. And if we take the outflow or the upflow, it is going into sinus number 1, it is going like this, it is going from here into the femur again. So this is a kind of channeling flow in the case of the legs of the orthopods and the hemipterans. So what are present for the pressure building mechanism? So we have got these compressor muscles which are present inside the legs for making some sort of pressure. So the leg hemocoel, it is partitioned into two sinuses, sinus 1 and sinus 2, and they have got a counter current flow mechanisms. So there is an opposite kind of flow as we have seen in the diagram previously. So the efferent flow in sinus 2, it doubles back at the tip where it becomes the efferent flow and it returns back to the thorax in the sinus 1. So in the sinus 2, it is going downwards to the tip of the leg, and then it is going upwards in the sinus 1 as we have seen in the diagram. So that was the case of the accessory flow or the accessory organs or the accessory hearts in the case of the arthropods and the hemipterans. Now if I go further, we have a case in the case of fishes, amphibians and reptilians. So here students, in these vertebrates, we have the lymph hearts. So this is the diagram which is showing you the lymph heart of a bufo. So it is of an amphibian. So these are contractile enlargements, I can say, of the lymph vessels. So these are the, you can say, expansions of the lymph vessels. So here you can see this is a lymph heart. So it has been labeled as the lymph heart. It has got ostia, many ostia, and these tend to force the lymph into the veins. So what it is doing, that this lymph heart will take the hemolymph into the ostia. It will enter into the heart from the ostia. Then the hemolymph, it will be pumped into the vessels. Now this is the hemolymph, which is this is the blood which is coming from the skin, because this is the case of bufo. So here we are taking the blood into account. The blood is coming from the vessel, from the skin. It is mixing with the blood from the lymph heart, and it is being propelled into the posterior vertebral vein, and these are the iliacs, or you can say the vessels from the iliacs, which are taking the blood also into this particular vertebral vein. So here the lymph hearts, they are the accessory or ampullary kind of parts which are present over here, along with the basic heart of the amphibians. So here the lymph enters the veins at many points and not through the thoracic ducts as in the case of mammals and the birds. So the lymph hearts, they are composed of striated, non-atomizing fibers, and they may have valves which prevent a backflow of the lymph. They are also present. So these are taking up the lymph and they are pumping the lymph into the various vessels of the heart. So it is a kind of another kind of a system which is parallel to the circulatory system. And here these lymph hearts, they are pumping the lymph part of the blood, of the circulatory system. So students, with this, we are finishing the types of the hearts on the basis of the morphology. So if I summarize this particular topic, then we have got 4 major types of hearts if we take the morphology of the hearts into account. The very first type was the chambered heart, which were discussed in the previous lecture. Now in this particular lecture, we have talked about the tubular, pulsating vessels, and the ampullary accessory hearts in different animals. So we have discussed the types, we have discussed the examples, we have discussed the diagrams. So you can, if you are getting small questions of suppose 5 marks on tubular heart, or you get a question on pulsating vessels, or you are having a question on the ampullary hearts or the lymph hearts, so you can give some examples with diagrams to comply with the question. Thank you very much.